the SITraN Open Day, which I know at least Greg came to this year, but I thought I'd bring them along because being on a Friday afternoon is not the ideal time for a lot of people to get over to Sheffield. So I've got on here my Twitter handle as well, so I tend to tweet on MND research, retweet other news. I'm also involved in a number of using genetics, not only in MND but other diseases, but I also do work. I'm sort of a hacker and a maker, and I actually made on this wall this motor neuron, which I'll get working in a minute, with LEDs to illustrate the signals going down and then failing in MND. So if you do happen to follow me and you get some random stuff, that's why. So who actually am I? There we go. It's the down button. Great, brilliant. So I originally started off in Sheffield doing my undergraduate degree in genetics. I then went to London and did my PhD down there before returning up north to Newcastle, where I joined the MND research group with Pam Shaw. So when she then moved down to Sheffield, I followed and I continued my career working in MND research, and in fact last year I celebrated 20 years. So I can certainly say in terms of progress from when I started, we have made a lot of progress, admittedly not as much as the patients and carers want, but the amount of research that is being done now compared to when I started is immense. It's gathering a lot more pace, and with some of the more drugs being used and trialled, hopefully some of those will actually now come through to the patients as well. So this is an outline of my talk. So it's on the genetics, but it's also emphasising why actually it's important. It's not just important to those that have a familial disease, it's actually a broader spectrum than that. And I'm going to talk about 3 different projects that we're sort of involved in, the last two of which are funded by the MNDA. So I'm going to start off with a bit of science, but don't worry, hopefully there won't be too much, but just to set the scene so you know why. So this is a stylized motor neuron, and the axon probably would go to the other end of the building. Bear in mind, your longest motor neuron is probably the one at the base of your spine that runs all the way down and wiggles your big toe. So that's over 1 metre long in a lot of people, particularly tall people. So, these are very big cells that have got are highly specialised, but in the middle they have all the DNA for all the information that you need to make heart cells, kidney cells, whatever. So that's what we're looking at when we're looking at the genetics, all that DNA. Unlike the English alphabet, DNA only has 4 letters though, A, C, G and T. And basically it pairs in this thing called the double helix, and you get C's paired with G's and T's paired with A. But basically that just means that when we're reading the sequence, we know, we can see how one strand works with the other. But basically the key message is that there's 4 letters. All the information is actually 3 billion base pairs of information is encoded in that DNA. And if you actually laid all that DNA in a cell and stretched it out, I know Greg's tweeted about doing the strawberry extractions, DNA extraction from strawberries at SITraN. If you actually laid all the DNA out in a single strand, it'd be over 6 foot. And all that information is packaged into this very small central nucleus in the middle of every cell. And you can see that double helix here, packaged all round into the nucleus. And it's found as these structures called chromosomes. And here, in humans, we have 23 chromosomes, and then males have XY and females have XX. We know that most of the MND is not found to be caused by changes in the number of the chromosomes or anything like that. It's actually spelling mistakes in the DNA sequence. So, spelling mistakes can occur naturally in your DNA, and that is actually what makes us have brown hair, blonde hair, blue eyes, green eyes. So there are variants in the DNA sequence that make us the individuals that we are. So for example, what we term the sort of reference DNA might be, I have a short-haired cat. It might have a change in another individual, may say, I have a short-haired rat. The sentence still sort of makes sense, but it is slightly different. Alternatively, you might have a short-haired bat, who knows? But equally, they still relatively make sense. So these are what we call variants, variations that make us all the individuals that we are. However, sometimes those spelling mistakes just don't make sense. So in this instance, a short-haired car. I'm not sure that. Or you actually miss a letter out completely and it's a short-haired CT. Again, it doesn't make any sense. And it's those sorts of mistakes that are what we're looking for in the DNA when we're looking in patients compared to controls without the disease. So when we actually look at the DNA, these are the results we find. So this would be a control individual. At this point here, there is a single peak, which is actually one of those letters, which is a T. But in the individual with motor neurone disease, not only have they got a T, but they've also got an A. So bear in mind you have one set of information from your mum and one set of information from your dad. So in this case, so mum has the T and dad is given the A, and that's what has then caused the disease. And you've got another example here. So when we're reading, we have to be very careful at looking very carefully at the sequence, because that one, they almost so overlay that you can't always see them. So you do need to, when you're analysing like this, look carefully. But as well as different, so in the same way, that there is where we've got the cat turning into a car. In this instance is an example where we're missing a letter. So in this reference sequence, you can see single peaks all the way along. In this sequence up here, it's single peaks, and then there's double peaks at every position. And then down here, it double peaks. And by comparing these two, we can see where that lost letters are. But why is all this important? So, going back, you've got all the information in the nucleus. Now, if you imagine you're an architect and you've designed a building, you've got all the diagrams, if actually the plumbers come in, they want just the information for laying all the pipes. They're not interested, where the electrical cables are in the walls. They just want to know where they're going to lay the pipes in the floors. So here they just take copies of the important stuff to them. In neurons and in every other cell type, they just take copies of the RNA that they're really interested in. So these are copies, and then the templates that make the proteins, and the proteins are actually the molecules that actually make our body work. Okay, and that's probably about as, I think, as scientific as it goes. But basically, a spelling mistake in your DNA means that the proteins that you're producing can then are then faulty and not working properly. So, as you're all more than aware, the majority of MND or ALS is sporadic with no family history. And so there has been questions raised to why we're spending so much time on looking at very much the familial disease. So basically, when patients go into clinic, there is no way to clearly distinguish, unless you start to take family history, between patients with a sporadic or familial disease. And so that means that we think there are similar pathways that are involved. So by understanding more about those rare genetic forms, the hope is that we can then target pathways that are then more widely applicable to everybody. So, and in terms of progress, so I actually started down here in 1996. So when I started work, we had one gene. We now have, I think there's 24 in here, plus another 2 which have been identified by the Project Mine even more recently. The size of the circles represents how frequent those variants are in those particular genes. And you can see in recent years how that is more plateauing with just more people, more samples, trying to identify those genes. And I think now, in terms of the familial disease, we probably have identified the variants in somewhere between 60 to 70% of the familials. What's challenging as well, though, is that sort of for patients and for the clinicians, is sometimes you can see these variants in apparently sporadic cases. So currently, it's not policy to screen everybody for the genetic variants, because actually in terms of treatment and diagnosis and prognosis, there is riluzole, and we haven't got yet that full information to give more details. But the idea is, as time goes on, we should hopefully be able to try and do that. So basically, in the majority of MND cases where there is a familial inheritance, you only need 1 copy of the faulty gene to get the disease, which means that generally offspring, it's 50%. But there are times when actually we know from reading the scientific literature that actually there are individuals who are in their 80s who are carrying large expansions in that C9orf gene and have no MND or dementia. And it's then like, do we need to then try and tackle those and see what's the protective factors within those patients? So yeah, so as I said, genetics, yes, it's important in familial disease, but we think it's also important in sporadic. So as well as those individuals where there are these variants, mutations occurring within apparently sporadic cases, we think that there's probably some risk factors within the genetics and within your genes which may make you predisposed, but only when you encounter an environmental factor. So one of the classic examples is the APOE4 gene. If you carry that, you have a higher risk of Alzheimer's, but carrying it doesn't mean that you will definitely get the disease. And we think that we know from twin studies, and in fact Ammar Al-Shaabi, who spoke at the AGM, has done a lot of work on this, from looking at twins, monozygotic with and without disease, they can establish that actually there is a genetic component, somewhere between 30 and 70%, even in sporadic disease, that gives you some predisposition. But as I say, it might be that if you don't encounter that factor, you can carry that predisposition and it doesn't have an effect, which in terms of genetics means that we have to try and tease things out, and it's a bit more challenging. So some of those environmental things, it might be carcinogens, hormones, a response to DNA damage, and these are all the sort of things that people are already working in the cancer field, and so as researchers we're looking and seeing how we can use the methods that they're using within our own field. So I'll now go on to talk more about some of those projects where we're actually sequencing the MND samples. So the first one is the 100,000 Genomes Project. So first of all, what is a genome? So it's all the DNA in the cell. Now the DNA isn't just in the nucleus, but there's also some DNA in things called mitochondria, which are little round structures which are actually where all the energy is produced in the cell to drive all the chemical reactions that the proteins do. It's important to know that actually mutations can occur anyway. I've spoken already about those spelling mistakes, changing the sense and making proteins not work, but actually the proteins, the DNA that encodes proteins only accounts for about 2% of your DNA. which is that little bit in red right at the end. Our genome is far larger than that. And what we do know is that the C9orf sequence is a repeat sequence in an intron. So we're now looking at not just that, looking at those protein coding regions, which is, to be honest, the easiest place to start looking, where we have done, but to start looking in more of the other regions and seeing if we can trace changes in the DNA that are more frequently found in patients compared to controls. So this is, the 100,000 Genomes Project is a NHS England initiative. It was launched by David Cameron in 2012. You may know that back in the late 90s, we started the Sequencing the Human Genome Project. So that was it was actually an amalgamation of 10 different people's genomes, but it took 13 years to sequence the entire genome at that point. Today, we can sequence a person's genome in a week. That's how far technology has gone, which means that this is all far more accessible for researchers to take things forward. And the Department of Health has formed this company called Genomics England that runs the project. And the idea is that once we've done this, that actually genomics and having your DNA sequence will be a regular part of your clinical workup as going for x-rays, having blood tests and things. That's the ultimate idea of going forward. So in the 100,000 Genome Project, they are also, it's across all diseases, so they're focusing on cancers, looking at tumour and normal tissue, but within the rare diseases, they're looking at the neurodegenerative, both at motor neuron disease and dementia. There is that's it, so it's run by genomic medicine centres. There is one in Greater Manchester. Basically, there are people, clinicians involved, and people can consent to have that those sequencing done through the consultant. The whole thing is standardised, and the whole aim is that rather than the DNA and the results just being held in Manchester, they're actually then open across England. So they're anonymised. In fact, let me go on to the next slide, no, next one. But so basically, I'll go back. They're anonymised, but that means that in terms of analysis, you can collate all the data, and in terms of this power in numbers, learn to work together. That's one of the underlying strategies of the 100,000 Genome Project. Everything is standardised for collection as well, so that actually whether a sample has been collected in Newcastle or Oxford or down at Southampton, it will have the same high quality DNA in use. So basically, there is quite an extensive consent process, I understand, but basically you are consented, they take the blood, they extract your DNA, and then they sequence it in a big centre down in Cambridge, and then they align all the sequences so that now this previous graphs I showed with the balls, that was the old style sequencing. This is new style sequencing, where actually you get 150 letters all in one sequencing pass, as it were, and then you have to all align them back to the reference, which is at the top there. And then the computer is able to say, this letter here in this patient is different, and it flags it up and feeds it back. Now actually you tend to get several thousand variants, because as I've already said, you have variants that change hair colour, eye colour, height, all those sorts of things. So it's then teasing out the differences that are associated specifically with the disease, which is why in the rare diseases, not only is it the patient, but where possible they're getting unaffected familial members of the family as well, so as they can work out which ones are, which the spelling mistakes are specific to the disease. So basically, obviously, as everything in the clinical, consent is key, but the samples are sent and anonymised to the sequencing centre where they can be looked at. When the results come back, they are de-anonymised so that the clinician can feed back to you. So this is very much a project embedded within the National Health Service and your care, rather than research per se. Although there is a sort of research element, it's very much a clinical project in the NHS. Okay, so I'll now move on to the Project Mine, which is now purely research. For the work that is being done with the UK samples, this is being funded by the Motor Neurone Disease Association. So basically the aim of Project Mine is to sequence 15,000 ALS patients from across the world, and they're also collecting 7,500 control samples. Then they will combine all that data together and then look for those variants that are associated more with disease. Now, because of the large number, as well as finding some of those variants that we know are associated with disease, like the genes we've already illustrated, this will hopefully enable us to start to find some of those modifiers as well. And so that, I've got a slide a bit later on where it shows that MND isn't all the same, and maybe trying to look at that data in light of those, we can then try and distinguish whether some of the basis of those differences are down to genes. So 38% of the DNA samples have been screened. In the UK, the MNDA have raised 87% of the target. They've recently been announced as being the charity of the year for Credit Suisse, which means that some of that funding from there will also go to this. A number of years ago, a large collection of samples were collected from the MNDA to form their DNA bank, and lymphoblastinoid cells were made from those, and so those samples are actually being sequenced as part of that. So that initial tranche and funding from the MNDA to set that DNA bank, they're still getting monies out of that to inform research. This is all the countries that are involved. So as you can see, a lot of European as well as Australia and I'm trying to think, USA down the bottom as well. So, and as part of the fundraising, it was announced last week that there's going to be a city swim. So Project Mine funding was originally came out of discussions in Amsterdam, and they've had city swims in the Amsterdam canals for the last several years. This is going to be in the Victoria Docks, and actually 500 metres is probably a shorter distance than colleagues have swum, including Amar, in the Amsterdam canals. But the funding is being split between these three charities. Okay, so on to AMBRoSIA. So there, my postdoc, Stephanie Sheffield, designed a poster for one of our research days, so afterwards you can feel free to have a look at that. But this is a multi-centre biomarker resource strategy for ALS. Basically, it's being funded off the back of the Ice Bucket Money Challenge. So initially, as I said, they collected all these DNA samples. Now they're collecting lots more samples and different sample types as part of this project. I'm just going to see if this is going to work, and if not, we will go. Let's see. It's not on the screen, unfortunately. Maybe if I...
The MND Associations' biggest ever project kicks off this September. More than 900 people living with motor neurone disease take part in this project.
I tell you what, well, that's not quite working. Technology. I'll see if I can get it up on the screen afterwards. But basically, there's a little bit of an introduction on YouTube, and what I can do is send Greg the link afterwards. You can also access it through the Motor Neurone Disease Association page. But basically,
Oh, God, it's not.
It's happens to the best of us.
It's usually me.
Right. Yes. Back on track. So basically, it's a 3-centre collaboration between ourselves in Sheffield, the University of Oxford, and Queen Mary, University London, along with the associated hospitals that feed into those universities. So the aim is to collect 900 patients and 450 healthy participants. And we are collecting blood for DNA and RNA as well, so looking at the copies that are being made within the blood. We are getting PBMCs, which is basically the white cells in the blood, so we're getting rid of all the red, because red cells actually don't carry any DNA. So we're getting rid of those. And also the serum and plasma. And they're just different components, and they have slightly different constituents. So it's a case of rather than looking at a whole blood where you've just got so much going on, by separating them out into the constituents, we might be able to pick something up that we can't if we look at the whole blood. We're collecting cerebrospinal fluid, and there's an illustration on the other poster about how we can use that and take the samples. Also urine, and also fibroblasts. So basically what it is we actually do a skin biopsy. So we take a little pinch of skin, and the skin goes down to the lower level so that the cells are proliferating, and we can then grow those up. And the key thing that we can now do is then actually convert those into neurons. So one of the other reasons why we used to focus again on the genetics and the genetic variants of MND is we had no way of modelling sporadic ALS. But now we can actually take the fibroblasts that derive from their skin samples and grow up neurons from patients that have the sporadic disease. And the main aim for all this is to look for biomarkers, and this is what the task is here with different coloured beads. So basically there are 4 different MND patients and 4 different controls, and by looking at the colours of the beads you then look to see which is the bead that is the marker of either the MND patient or the control. And we're doing that in a lot more complex environment. But it gives you the idea, it's not always just that this protein or this microRNA or this RNA is present or not. But that's you can have a look at that after the presentation. So this is what I was alluding to. This is actually a slide that I got from Brian Dickey of the MNDA only yesterday. But I thought I wanted to include it because it clearly illustrates that actually MND is not just all the same for everybody, as you will be able to tell me better than most. You've got about 20% that are quite slow, 60% is sort of more the classical progression, and then about 20% of the progression is much faster. And it might be that actually some of this is down to genetics, and it might be that within these groups, different drugs will work better than others. So trying to actually stratify and be aware of the differences in the population of those with MND, and trying to stratify when we're doing the drug trials will be really important going forward. That's what it is. So I had this is a bit of light relief. So I gave you the AMBRoSIA use of acronyms. This was a PhD Comics tweet about how actually you can make up, you actually sometimes start with an acronym and then try and fit the text around what you actually want it to be. And to be honest, a lot of projects now when we're writing grants, you have to have an acronym as part of the procedure. So with AMBRoSIA, we wanted to do some sequencing of those samples so that we knew which ones can change genetic variants in DNA. So because it's AMBRoSIA, we came up with NECTAR as an acronym, which sort of fits. next generation genetic screening of the ALS biosample repository. So, but it sort of fits. But basically what we are now doing is anyone who is recruited to the AMBRoSIA study in Sheffield or will then have their, for research purposes, their DNA sequenced for the known ALS genes, so that when we're then doing research, we can find whether there are biomarkers that are specific to one genetic subtype. It might be that if you wanted to do some research, you can just do it on those specific samples. And it may also help us establish why, say, a variant in a gene that's dysfunctional in every cell in your body only causes your motor neurons to die. And this is also funded by the MND. So that's it. I just wanted to make sure that I wanted to thank everybody, because I'm sure at some point you've all taken part, you've done fundraising, you're raising awareness, and as someone who has been in the field for a long time, the body of support has also increased, as well as all the research, so thanks a bunch. Any questions? Yes.
You talked about twin studies.
Yeah.
So my twin brothers behind me here, one with
Yeah.
MND, one without.
Yeah.
Identical twins, so they have the same DNA apparently.
Yeah.
Is that right? You know, how is that going to be developed anymore, or have we kind of just come to an end with that?
No, I think we are interested in collecting the DNA where that does happen. So this is where, so your DNA is one lot of information. What can then happen is that you also have modifications to that DNA which are environmental, and that is then called epigenetics. So that's one thing that they're looking at, where there's twins, and seeing what the differences are, as well as whether there are any variants. We would expect it to be identical, because that's where, but it might be on all the times, bearing in mind you come from sperm and an egg, and then there's multiple and multiple and multiple, errors can happen. So is it that actually an error has happened at some point during that process? So, no, collecting twin studies, and certainly I know in Holland they're actively doing that, but we have had a number of DNAs from twins.
They have had it taken.
You have had it taken?
So Dr. Cooper,
Oh yes,
yeah,
I know John. I supervised some of his PhD. So yeah, so that will be part of a study that we will then certainly have a look at. So.
Can you elaborate at all on the SITraN trial that's happening at Sheffield, and potentially the C9 North 72 one that may happen in the future? Is there any more things on that sort of?
The short answer is I have less visibility on that, because that's more the clinical trials side. So no, I know that there are studies going on elsewhere against the C9orf72. So basically this is where we know that there's the faulty protein, but actually they're specifically targeting. I was saying about you have these A's pairs with G's and A pairs with T, C pairs with G. You can actually put stuff, an oligo, a bit of DNA into the cell and stop copies being produced. And it's called antisense oligomers, ASOs. But basically, it's a way of trying to stop those faulty proteins being produced. And they've started it in SOD1, and they're also looking at it whether it's then can be used in C9orf72 with this huge expansion. So I know there is work ongoing, but unfortunately I'm not aware of up to date with how things have gone in Sheffield at the moment, other than it's ongoing. I will be around, so if you want to have a break, you can speak to me or whatever. I will try and get the motor neuron going as well. Hopefully my clock's got some charge in it.
Get your presentation from somewhere to show the people, obviously there's only me from my family here, and I'm not going to do that.
Yeah, I don't have any problems. So what I can, if I put it on the laptop,
Yeah, sure.
and then if you want to email. Obviously, Greg has been recording it.
Yeah.
Oh, yeah, okay, that's fine.
But whether the slides have come out, what it might be worth doing is having the slides as well, and I'm happy for you to have that.
Thank you.
Okay.
Anybody, any more questions? No? Well, thank you very much, Janine. That's a fantastic presentation. It's always nice to know what's going on in the research world, and it's always nice to have someone who can get it across to us. Because we do occasionally, there have been, not in our branch I've got to say, where it goes whoosh straight over the top. But yeah, fantastic presentation. Thank you for coming.